So welcome back. For any of those who have missed the first lecture and are joining for the second, so this is part of a 2-hour symposium block, which really emphasizes kind of model organisms, the fruit fly, and the history of genetics in the 20th century from the historical sociological perspective and the data, and now with Thomas Stoeger, the data science, modern contemporary perspective, with the idea that both perspectives are mutually informative and mutually beneficial for the advancement of both inquiries. So thank you again for coming, and I introduce without delay Thomas Stoeger. Thank you very much.
So welcome, thank you for being here. Perfect. Anyways, so in the next roughly half an hour, you will hear and you will see about the model organisms and their impact on human research. But as you can anticipate from this first slide, the scheme of what we try to do extends beyond model organisms. This talk will be separated into 4 parts. The first one will be dedicated to viewpoints. You will learn about my biases and a system which I believe can be very helpful for biologists, but also people from different disciplines to better understand distinct biases within biomedical research. The second talk, part of the talk, will be about influence. And we will rediscover the influence of model organisms on research on human genes. Then we will take a full stop, and you will see and hear about ossification. And you will learn more about this later. Finally, in the last 3 to 5 minutes, you will see one example how meta-science can inform specific biological inquiries within a lab. Let's start with viewpoints. This is a picture I took as an undergraduate student on the fly. Just before taking this picture, I was literally kind of shaking all over my body, and I asked lab members to help me take this picture. I was very nervous, running up and down, and very exciting, because this gene, or this mutant, lies in a gene that encodes for a completely uncharacterized gene. Those of you who have maybe looked at some of these wings before will immediately recognize that this is not a typical shape of a wing of a fly. It's not elliptical. Instead, it has these kind of notches. And mutants in genes that lead to these notches lie in the so-called notch pathway, a signaling pathway that's important for animal development, but also in some kinds of cancer. So you can imagine a series of those mutants, new genes involved in the notch signaling pathway. But let's fast forward 10 years and visit Flybase, an excellent computational resource. Essentially, Flybase accumulates all types of different information which is known about individual genes in the fly. And here is the summary of one of those genes. And every green square that you see indicates that something is wrong. You do not indicate several of them. In short, this was the impact of our publication. We had roughly 300 citations, but no single other researcher would follow up on one of the newly identified genes that we had found. These genes I would be so excited about. And to me, this indicates that biologists just get close to that. Sorry. Sorry. Oh, yes. Oh, yes. No. And to me, this indicates this kind of discrepancy between kind of citations, number of citations, and follow-up stories, that we are missing out on something important in understanding how biological science works. Taking one step back again to Flybase, you can think of a system that would be very nice to study how science works. And this brings us to a few points. Ideally, we want to have a framework where we get all the biological information that's out there. We are presently integrating all high-level bioinformatic resources that exist on human, but also some model organisms. We try to be comprehensive. We also study products and literature, the public literature that's out there on genes. Also, we're now collaborating with 2 publishers, and we're also tracking individual citations between every paper to another one. Other readouts on products would, for instance, also be genetic tests or clinical trials, basically anything you can think of. We also want to study scientists. So we track every single scientist who has ever published a paper in a disambiguated manner, also tracking their career and what they have done before. Society. We track all the public funding, and we track some other things, such as for some journals, we're now starting to move towards the reviewing process. Or a lab mate of mine got into some problems with publicity when collaborating with Dropbox to actually study how scientists share individual files. And we are also trying to track technology included in an individual paper, and technological possibilities that are out there, because all of those things put together shape the way we produce knowledge. And really, all of those things belong together. And what we have been building was kind of this framework where we can combine all of those many different sides of science. With this, we move to one example, influence. And we will discover model organisms. But we start with this observation on human genes. And these are some of my most favorite publications. Essentially, only a subset of human genes are actively studied. You might have seen a graph similar to this one. So this is looking at this PubMed database of biological knowledge, literature. We're counting for every protein coding human gene in how many publications it occurs. We see that some genes occur in hundreds of publications, other genes only in a handful. So if we would gain a sufficient understanding of the processes underlying biological knowledge formation, we should be able to put this into a model. And this model should be able to tell us exactly why a certain gene is studied heavily and while another gene is rarely studied. And through these models, we can test different assumptions. For instance, we might want to start with chemical, physical, and biological characteristics and phrase them in a machine learning model. For instance, we can take 430 very generic features describing genes, transcripts, or proteins. Then we take a random subset of those genes, place it in our machine learning model. Then we go to another set of genes and apply this model to ask how much can we actually explain, for instance, in a number of publications, through the features. This is a quantification, and the following slides will all have a similar setup. On the left-hand side, you will see different assumptions that we test. On the right-hand side, you will see a scatter plot. On the x-axis, we have on a logarithmic scale the number of observed publications per gene, and the y-axis the predicted one. And you can see that with these characteristics, we can, to a large extent, quantitatively predict the number of publications per individual gene. For the sake of this talk, I'll also add that is a small part of a pilot that we did, which was on the fruit fly. So the whole scheme does not only work on humans, but it also works on other organisms. But let us return for a moment again to human. This correlation is high, but it's not perfect yet. So we were wondering whether we might miss out on something. And to better understand whether we might miss out on something, we wanted to at first know what are actually the characteristics that drive the number of publications in human genes. So we could take these models and unfold them. Here you see in different rows independent randomizations, and the columns mark different features. And the importance of those features to our machine learning models is shown from white to purple. And you can see that only a small subset of features, namely 15, drive most of this explanatory power. Many of those characteristics are things that we might anticipate, such as the sensitivity to loss of function mutations, or the fraction of tissues in which a gene is active. There are also other properties, such as the presence of a signal peptide that mediates the transfer of proteins out of the cell, or protein gravity, which is a measure of the hydrophobicity of proteins, or the length of genes and transcript molecules. But the important thing about this is that this is a very generic list of biological characteristics, which is very small. And yet this very small set explains a lot of what we're actually doing. Another thing that is important is past research. So we hypothesized that taking into consideration also the year of the first publication of a gene will further explain the number of publications, and this is indeed the case. Yet it's still not perfect, as you can see. So we were wondering whether we might be missing out on something, and whether these first publications could give an indication about what we are missing. And you already know that model organisms will now be coming. Looking at the first publications on a human gene gives a good reference, but we also looked at all of the other publications on human genes. And then we looked at the publications which are cited by those publications. And then we analyzed the content of those publications, and in short, we found an overrepresentation of model organisms. The first publications on human genes are twice as likely to cite a publication containing a gene of model organisms, the most prevalent mammalian one being mouse, and the most prevalent non-mammalian one being fly. So we're wondering whether citations indeed indicate influence. And if we take these related autologous genes of model organisms, we do indeed see this. And this slide is terribly complex, but the only thing to remember from this is that the one on the right-hand side gives a much nicer diagonal line, indicating that if we consider the year of an autolog, we can accurately predict the year of the discovery of a human gene, whereas if we only consider the features you've seen before, we are not able to do that as well. Similarly, for the number of publications, if we introduce the year of the first publication, we can again increase our ability to predict the number of publications on human genes. And if we consider the number of publications on related genes of model organisms, this gets very close to 0.9, indicating that now we actually have a very strong ability to predict quantitatively at the level of single genes, by single genes, actually started giving a good baseline. So to summarize this first part of the talk, you can test quantitatively mechanisms behind the formation of knowledge by feeding different assumptions to a model. For instance, you can take biochemical features, and some of those are very important. We can test the influence of model organisms, and we do indeed find this influence of model organisms that has at least partially acted through shaping these initial publications on human genes. And together, we can recapitulate quantitatively many aspects of science and predict many parts of it, such as the number of publications. But we can also predict other things. For instance, we also once tried to predict NIH funding, and all other things you can relate somehow to genes. But there's a problem. Somehow this does not resonate well with this idea that I told you in the very beginning, namely that there was like no follow-up on our study. And this indicates some process, something must have changed somewhere along the line, and that is ossification. Ossification means that scientists would largely stop to explore new areas of their domain and rather strengthen the things which they already have some indication of. And there's absolutely no reason to believe that this is specific to genes or to biology. Others have found that in a couple of hundreds, or even I think thousands of scientific disciplines. So when scientific fields grow, and once they reach a certain size, this seems to be almost inevitable. And the reasons for these ossifications are still not completely understood. It could relate to the capacity somehow of a field. And I would love to tell you exactly what would cause ossification, but I think we're not exactly there, so you will now also see some preliminary results. But let's first look at ossification in a little bit more detail. You've already seen this slide. These publications have been that mark this finding have been published over several years apart. Here we'll have a scatter plot comparing for each dot being an individual gene, the number of publications in the years up to 2010 to 2011 to 2015, and they're largely correlated. Only around 6 genes strongly deviate from the general distribution, meaning that they're much more studied than anticipated by past trends. And as you might know from this institute and the Cancer Genome Atlas, one of them nice findings from the Cancer Genome Atlas is this particular gene, IDH1, which indeed defeats this general trend. So meaning if there's actually very strong evidence, things can still move up, but this number of genes that strongly changes from past research patterns in absolute terms is very small. The next slide will show the Gini coefficient. The Gini coefficient is a measure of inequality going from 0 being perfect equality to 100 being perfect inequality. Things that are generally higher than 60 or 70 would be considered massive inequalities. And we will see this for human genes and the distribution of publications on individual human genes, going from the 80s to now. And what we can see is 2 things. First, research has never been as diverse as it presently is, which is a really good thing. But also at the same time, we see that one point during the last decades, there was this time where biologically grew and probably went over this threshold of ossification. And since around the year 2000, the research does not diversify much further. So what contributes to ossification? Well, we don't know for sure. And there seem to be many things that are able to contribute to ossification. One of them you will see now in this talk about model organisms relates to model organisms. And more specifically, you will see that the share of model organisms on the overall research that's being performed has been decreasing. That not only my study, but also other studies have not been continued. And that the job prospects of people that nowadays work on Drosophila are actually lower than, for instance, for human, which probably could drive some feedback mechanism. But there probably is more. And in this video, you will see the development of citations between different organisms. The thickness of the lines is proportional to the total share of citations. We're starting at 1980, and you see a lot of the citations relating to genes go from E. coli to other E. coli. And as we will move on in this video, you will see at the bottom right this concentration towards human, rat, and mouse. So let's look at the last 30 years unfolding. We really see that there's kind of this change in this landscape where model organisms get a smaller and smaller role. Where now basically most of the research on genes is done in humans and in mice, which are systems that do not lend to like some undergraduate mutagenizing hundreds or thousands of animals and then assessing the phenotype. No continuation. To assess whether we were the only ones, we also looked at a list of roughly 150 screens done in fruit flies or in cells of fruit flies where individual genes would be mutated. And we asked whether actually these genes would then also be cited by other studies. And you can think of this like this. So in fly, something that people like to do is to do so-called screens, where they have a set of genes, and then they mutagenize them. And sometimes they find something like this that doesn't look normal, and they call it a hit. So we were wondering if now we have a small-scale human study, which actually cites this fly study, and we are only looking at genes that are evolutionarily conserved, to which extent do they actually study genes that have been studied in the initial fly paper? And here's the summary. Essentially, for only roughly 13% of the publications pairs between a fly paper and a human citing paper, there's at least one or possibly even more genes which are shared, meaning that actually most of the time the citing publications, they do not study the same genes. But there's also a special case where maybe they contain a gene, but it's not a gene which the initial authors found to be interesting. And reassuringly, we find that this fraction is much lower. But jumping back to the beginning, you will recall this graph. There's this large inequality in the number of publications per gene. So if we now focus on the kind of this left-hand side of this distribution in genes, which are essentially unstudied, you will see the following thing. If you see these little studied genes, even if they were identified in the initial study to have some interesting phenotype, it's only around 4% of these pairs which actually follow up. This means that for these little studied genes, it's essentially as likely to be kind of cited as for the publications on a heavily studied gene, even if the heavily studied gene did not show anything interesting in the initial report. So this leads to the very last part on the job prospects. These are is looking at the transition of first authorship to PI-ship in the last decade. This is for humans. This curve decreases, which partially is because people are still in training. But we can see now that, as maybe anticipated by kind of this decrease of research on model organisms, that doing a postdoc or PhD on flies will actually mean you will have a lower job prospects. And if we correct for things like duration of training, this would get even more massive, this effect. On a related note, we find that people that work on flies nowadays in this time span, that they actually rarely move to other model organisms. Kind of now looking again now in these early years of being a principal investigator, you will see here the 10 most studied model organisms. Fly is still on position 4, and only roughly 40% of the fly people also move to some other model organisms as a junior investigator, meaning that most likely they will be for a long time working on flies. So to summarize this up, we find that there has been this shift away from model organisms. We see that new things from large-scale phenotypic assays in flies are presently not followed up. And we see that nowadays there are lower job prospects for people that work on Drosophila as compared to other systems such as humans. So if we talk about the fly, I think we really have to separate between two parts. One is the influence prior to this ossification on research on human genes, where we find strong evidence. And then there's at these more recent years where the influence of model organisms, at least in terms of opening research on new genes, has been steadily decreasing. Finally, for the information, the system that you have seen could be used in many different ways. For instance, it could be used for some bioinformatic analysis such as this. Maybe anyone has an idea what this could be. So these are the genes most studied by researchers funded by the NHGRI. And on the individual rows, we have individual experiments in Drosophila. And it will be white whenever these people in Drosophila have identified this gene to have a different phenotype in their screens. But there's also a more personal example. I'm interested in gap of knowledges. And one area that seems to include many parts of biology is aging. And now I'm presently also leading some analysis on a kind of consortium work on aging. And more specifically, I'm still interested in gaps of knowledge. In short, if we do monitor transcripts across all types of organs of mice, we find with statistical significance slightly over 8,000 different genes to change with age in at least one tissue, which is a large number. However, I could also develop a very rigorous statistical approach to maximize reproducibility, which led to a small set of 110 genes. And now comes the meta-science. Having this framework, we could do an inverse literature search to essentially look for everything that has not been linked to aging, which yields certain genes, which with very high empirical evidence are linked to aging, but nothing can be found in the literature. So we decided to follow up on one of them, and it's a gene that regulates the transcriptional elongation. And we find the following observation. On the y-axis, you see the fold change in the transcript levels between individual ages. If it's above 0, it means that this gene becomes more active in older animals. And if it is below 0, it means that this gene becomes less active. So we still have somewhat this problem that many genes seem to change, but often the effect doesn't seem to be that strong. Having this insight from the inverse literature search and this specific candidate gene, we looked at the length of the gene. And what we find during aging, and we find that in 3 out of 4 different tissues and in several other model organisms too, that what's happening in aging is actually that as we age, there's this gradual imbalance of transcripts encoded by short genes versus transcripts encoded by long genes, messing up a lot of the things that happen in the cell. But I really want to stress out that this is kind of one example of applying concepts from meta-science to very specific biology. And with this, I want to thank you and summarize everything. So there is this viewpoint. Everything, I think, belongs together. We can measure influence of model organisms. There has been this ossification of biological research, and information is something that could be used. And in a way, all of this also describes part of history of science. And flight people also like to share, and the same is true for us. If you're interested in some of that, most of this framework is quite accessible. It's written with data scientists in mind. Just reach out to me. There's some legal restrictions for sharing some part of the data, but the system is very much set up for sharing. And I would like to thank my co-workers and also my mentors, Luis Amaral, Enrique Moimote, and Northwestern's Data Science Initiative, that really allowed me kind of to work on a topic on which I had like no background on a topic that might actually fail, which still was very enjoyable. And thank you. I'm looking forward to your questions.
Questions, and as a reminder, if you have a question, say who you are, where you are on campus, or if you're not on the NH campus, say you're not on the NH campus after saying who you are. So questions, comments? Yes, the gentleman.
So I think one problem is whether it's fair actually to recommend junior investigators to do something else, because in a way, it also means suggesting them to do something that might be bad for their career. So I think it is some kind of more complex system which probably need to involve different parties and some type of kind of protection for junior investigators that move to other areas, and also making people more aware. Maybe potentially with this kind of framework, we could very much pinpoint of who maybe would be doing some biology close to that, and who could maybe profit by like some collaboration with people from different fields. And probably on a global level, probably it needs to be like, I think, some type of protection and a small one recommendations. But the truth is we actually do not really know, because we presently have not any empirical evidence for any of these interventions.
Question up there?
Yeah. I think that is something that could potentially be done. I'm presently only aware of one other research group outside of NIH that has access to the funding decisions. We don't have that access. But probably it's certainly something that could be done.
Yes?
Yeah. This is citations, yes. I think the citation is more an empirical observations, that there's like less research done on model organisms. And the research that is done on model organisms mostly is on mice and rat. So I want to add that also, like, for some other things, not only like for you, specifically for humans, but in general, if one would, for instance, look at kind of funding from the NIH versus other organizations, like everything that maybe doesn't have NIH on the grants, people still seem to kind of be studying similar things. So I think there's actually like some limited flexibility of the general funding schemes.
Further questions? Yes.
Some related issues, like that certain subfields of biology, like I'm not sure if that maybe has changed in the last few years, but like from some more archival data, what we published is that it can also lead to other inequalities. Point. We haven't tried that yet, but so based on what others have published on this kind of general process, one hallmark is that people start citing older paper more frequently. So potentially one could imagine that if there's like this shift between older and newer publications, and if that maybe happens in a subnetwork, which cites each other and hence might be a subfield, one would probably be able to formulate something on the network field thing. So that could actually maybe work out quite well if a field generally all of a sudden shifts to kind of not citing newer stuff so more. There are many excellent things to be said about the genomic perspective. I think their main argument comes like from something outside of biology. If it's a more general process, probably biology also adheres. Other argument, potentially paradigm shifts, as classically defined, would be something that's considered to be relatively rare, and sometimes kind of happening after some time where people felt that something just kind of wasn't right, and maybe also actually more prone to accepting some things that did not entirely fit. So I think this kind of preceding development seems to be somewhat different from the transition to genomic, where a lot of people said that this actually makes a lot of sense.
Any additional questions, comments? So I have a couple of observations. The one thing that's kind of interesting, and this goes to the paradigm shift, is that we really need to rethink a little bit scientific knowledge change, because paradigm shifts assume that the two paradigms actually can't converse. So whenever people discuss paradigm shifts, at least in the traditional Kuhnian sense, and Kuhn changed his mind constantly, which was his problem. There's lots of approaches that are seen as paradigm shifts, but in science. But if they can talk to one another, they're not considered paradigm shifts, at least in the classical sense. The other thing is I never thought my reading of Corrado Gini would have anything to do with what I do here. But so Gini's coefficient comes out of his. Gini was an Italian sociologist who wrote, I think actually until the 1950s, and he was, interestingly enough, there's a huge literature on what you're talking about, which is why certain things are so. What happens in elitism in science, whether it's why people keep winning Nobel Prizes if they work in Nobel Prize-winning laboratories, and why certain genes get all the attention and all of this. So what's interesting is that I think maybe what could be some future for the model could be there's a lot of very technical work in sociology and social science about elites and about ossification, or as political scientists call it, incumbency. So why do we have this display that doesn't really quite work very well? It's because of incumbency. We're used to it. So a lot of things can be explained. A lot of that missing sort of, the missing part that you're looking for is because scientists and people generally are very comfortable with the keyboard that they're using or the model system, and that's just human nature. But there's a lot of, so we should talk later, because I actually think that in a certain way that social processes are probably better known than biological processes, and that we know more about sociology than we do about biology and genetics, even though we've made some advances. So there's maybe some of that could be helped by really looking at, which is a claim that I'm not gonna back up. It is a claim There's a very big literature on elitism, incumbency, ossification, disciplinarity, these types of social phenomenon that I think would be really fascinating.
Also, I think like one very beautiful thing about biology, at least since the Human Genome Project, is we kind of know about the genes, so we kind of have an idea of the things we don't necessarily know much about, which is actually kind of, we can think of it as kind of a blessing too. It's not everyone kind of has something like that.
After I made a totally indefensible claim, are there any other further comments from the gallery here? Okay, thank you very much.
Thank you.